In this lesson, I would like to address a little bit about bacteria and then viruses, and specifically some information that you need to know about them so that as we begin to study recombinant DNA technology and some of the new biotech techniques, that you can understand kind of what we're talking about, because we use a lot of bacteria and we use certain viruses in some of the biotechnology that we do today, and so I want you to understand a little bit about them before we embark on that. First, remember, and you probably do remember, that bacteria are prokaryotic cells. They are not eukaryotic cells, which means they do not have a nucleus and they do not have any membrane-bound organelles. So you're not going to find any ER or mitochondria or chloroplasts or Golgi apparatus or vesicles or vacuoles or anything like that, anything made of membrane. Their DNA is different. They don't have chromosomes. They have a circular piece of DNA that's found in a region called the nucleoid region, just sort of within the cytoplasm. They do have a cell wall and, of course, a cell membrane, but their cell wall is made of different material than plant cells, which shows you they're not terribly closely related to plant cells, although they do have a cell wall. Now, we need to learn a little bit about bacterial reproduction. Under most conditions, bacteria will reproduce through a process called binary fission. It's not like mitosis. It's very different because, of course, they don't have chromosomes. In this process, binary fission, the DNA is replicated. It happens a little differently than it does in eukaryotic cells because there's no chromosomes, but a piece of the DNA attaches to the membrane and then is duplicated as it's attached there. Now, it's going to be attached to the membrane because, of course, as the bacterial cell splits, the DNA has to be separated into the two new cells. So you can see here that the duplicated DNA is attached to the membrane. The cell eventually elongates and then splits in half, and you get two new daughter cells. Again, they're clones of the parent cell, so this is a form of asexual reproduction. Now, bacteria do undergo some forms of sexual reproduction under certain circumstances, and there are three kinds. The first kind of sexual reproduction in bacteria is called transformation. This is a term used whenever there's an exchange of DNA between bacteria. So it's sort of a general term, and it's used generally when we're not sure exactly how that transformation happened, but there's some way that information from one bacteria got to the other in terms of DNA. A more specific term, and another type of sexual reproduction that we talk about in bacteria, is bacterial conjugation. In conjugation, certain bacteria can actually swap a piece of their DNA with other bacteria. It's only certain bacteria that do this, that do the giving and the taking, and so not all bacteria can do this. They're only the ones that have this extra piece of DNA called a plasmid. Now make sure you write that down, because that's going to be important later when we study about recombinant DNA technology. These plasmids are going to be important, but only certain bacteria have this little extra circular piece of DNA called a plasmid, codes for like just a gene or two, something very specific, and those bacteria that have them are able to reproduce through conjugation. The way it works is there's a duplication of the plasmid. At the same time, there's a conjugation tube that's formed between the two cells. This piece of plasmid is inserted from one bacterial cell to the other, and then again, it's duplicated. So the resultant two cells that are after the conjugation, of course, you start with two, you end up with two, but you have the passing on of this plasmid from one cell to the other. You have, notice, an old piece of DNA combined with a new piece of DNA, so it's semi-conservative in the way that it's replicated through that process. That's called bacterial conjugation. And then the third type of sexual reproduction in bacteria is called transduction. In transduction, bacteria are changed through a transferring of some DNA from a virus. And I think we talked before about bacteriophages. You saw, I think, a video about bacteriophages. These are specifically viruses that infect bacteria. In this case, some of the donor DNA from the donor bacterium gets incorporated into the viral particles as they reproduce themselves. And so you end up with, when the bacteriophages infect another bacteria, that piece of DNA is inserted into another bacteria. So it's being transformed, but through a virus. A virus is actually doing the transferring of the DNA. This is called transduction. Now moving on, let's talk a little more about viruses. Just to remind you, they are not alive, so they're technically not living organisms, although they interact with and reside inside of other living organisms. There are strains of viruses that can infect just about every living organism. So we think of viruses that infect humans, but they infect plants, fungus, they infect bacteria, as we just saw. They infect every living organism on the planet. What are they? They're basically a piece of nucleic acid. They might be DNA or RNA. And then they're surrounded by something called a protein coat. So it's just a structure made of protein surrounding a piece of nucleic acid. So they're not made of cells. They don't have a metabolism. They don't reproduce on their own. They cannot reproduce on their own. And so because of these reasons, they are not considered alive. Now, as I said, a virus can either be made of RNA or it can be made of DNA with a protein coat around it. Those that are made of RNA are called retroviruses, which means that in order for them to make copies of themselves in another living cell, in a living cell, they have to actually transcribe backwards. So in other words, remember, transcription is DNA being copied onto RNA. They need to copy the RNA back onto DNA in order for the cell to make more copies of them. That's how they reproduce, by getting cells to make more copies of them. And so because of that, they have a special enzyme found only in these viruses. They have the ability to get the cell to make this reverse transcriptase enzyme, which causes a reverse of transcription, and DNA is made from the RNA, and then the cell incorporates that piece of DNA into its own genome, and it is copied along with the cell's DNA, and that's how the virus reproduces. Here are some examples of some retroviruses. Polio, you may have heard of. Cold and flu viruses are both retroviruses. Ebola, you may have heard of, and then HIV, the virus that causes AIDS, is also a retrovirus. The other type of virus is made of DNA, again with a protein coat around it. DNA viruses simply insert their DNA into the host cell, and then they insert that DNA into the host genome, and then it's copied along with the host's DNA. More of a straightforward process. Examples of DNA viruses: smallpox, herpes, the virus that causes warts, and then chickenpox virus. These are all examples of DNA viruses. Here are some electron images of some viruses. Again, these are much too small to see with our compound microscopes that we have in the classroom, but with an electron microscope you can see a very, very simple structure, a lot of them kind of round. This one here is more of let's see, a hexagonal shape, I think. Maybe it's got 7 sides. No, it's hexagon. So they have very strange, they kind of look like spaceships sometimes. And I want to finish up by talking a little bit about viral reproduction. There are 2 ways that we think about viruses kind of living out their life. Even though they're not alive, we kind of think about them having this life process. In the first form of the viral infection, this is called the lytic pathway. In the lytic pathway, the virus enters the host cell. It quickly reproduces more of the virus. So it takes over the host machinery, gets the host to make a whole bunch of DNA and protein to make more virus particles, and then the new virus particles burst out and kill the cell immediately. So lytic as in lysis. You've heard of lysis, I'm sure, means to split open, and so these are viral particles going in and completely taking over the cell and just killing the cell and bursting out and infecting more cells. This diagram illustrates that process. Here we have, in this case, a bacteriophage coming in, attaching to the host cell, inserting DNA here, getting the host DNA to the host cell to make a whole bunch of virus particles, and assemble them together, and then eventually they are all ready to go. There's lysis and release. The cell splits open and all the particles come out. This is called the lytic pathway. The second form of lifestyle that viruses have, it's not really a lifestyle because they're not alive, but you know what I mean. The second form that they have when they infect cells is called lysogenic. In the lysogenic pathway, the virus enters the host cell. The viral DNA gets incorporated into the host cell as before, but in this case, the viral particles just get copied along with the host cell DNA almost indefinitely, and later they can enter the lytic cycle and burst open, but in the meantime, they're just sort of incorporated into the host cell DNA and copied along with the host cell DNA for long periods of time, potentially. So here you have, again, the virus particle attaching, the DNA being inserted, it gets incorporated here into the host DNA, it gets copied along with the host cells, and through generation after generation is just incorporated into the host cell. This is called lysogenic. Now, in many cases, viruses may spend part of the time in a lysogenic pathway and later are activated to undergo the lytic pathway, and this is not uncommon. Last but not least, almost finished, there are some other infectious particles I just want you to be aware of. These are smaller than viruses. Again, they are not alive, not living. These are just particles that infect and cause disease in other living organisms. You may have heard of viroids. You may have heard of prions. Viroids are small pieces of RNA without a protein coat. So they're just a piece of loose nucleic acid, and they can infect other living things and make them ill. The most common are seen are plant viroids. So they most commonly infect plants, although there is a viroid that causes hepatitis D. Prions are really weird. They are just a small piece of protein, and these are not proteins that are not already manufactured in a lot of cells. So these are, just a little bit different enough to be prions, but these are proteins that have been seen in other cells. They're altered gene products, though. What they do is they change the shape of the protein from a helix shape, which we talked about, to the beta pleated sheet. And this change, as it attaches to cells, this change can cause clumping and a buildup of the protein, and most commonly it's seen in nerve cells, in brains. This is the infectious agent that causes mad cow disease. You may have heard of that, or BSE stands for bovine spongiform encephalopathy. It also causes prions also cause a similar kind of disorder that's seen in sheep called scrappy. So that's it for bacteria, viruses, and other infectious particles.